Hello everyone, welcome to Smita Kiran Biology classes. In this video, let us discuss transcription and the structure of transcription unit. This topic is very important for theory exams as well for board exams. From earlier topics, you have studied that Watson and Crick, after the proposition of DNA double helix, have proposed the central dogma of molecular biology, according to which DNA, when it undergoes the process of replication, produces DNA itself. And the same DNA, when it undergoes the process of transcription, produces mRNA, and upon the process of translation, mRNA produces proteins. Now, during the process of DNA replication, entire length of DNA, how much ever be the DNA is, it get duplicated. For example, in Escherichia coli bacteria, the length of DNA is 4.6 into 10 to the power of 6 base pairs. You know that the length of DNA is measured in terms of number of nucleotide base pairs. So, the entire length of DNA is replicated, and you should remember both the strands of DNA will act as a template. So, there is a synthesis of two another molecules of DNA which are complementary to the parental DNA template strands. Whereas during the process of transcription, only one segment of DNA undergo transcription. That too, only one strand of DNA will act as a template. So note this point, very important. Only a segment of DNA, here I marked, only segment of DNA will undergo transcription. That too, only one strand of DNA act as a template, that is when DNA is undergoing the process of transcription. And here DNA dependent DNA polymerase enzyme is responsible for the transcription, whereas in replication DNA dependent DNA polymerase, here DNA dependent RNA polymerase is responsible for transcription, whereas for replication DNA dependent DNA polymerase. Now this DNA dependent DNA polymerase can polymerize the RNA molecules in 5' to 3' direction. So it is similar to that of DNA replication itself, but here adenine is complemented by uracil. So you should compare the things now. During replication, in template, if there is adenine, the complementary strand will have thymine. If it is thymine, adenine, guanine, cytosine, cytosine, guanine. Whereas during the process of transcription, if the template have adenine, the newly synthesized mRNA will have uracil. If it is thymine, adenine. If the template has guanine, it will have cytosine. If the template has cytosine, then it will have guanine. So that is the thing which happens during the replication and transcription respectively. So you should note these 2 points. During the process of transcription, a complementation of uracil instead of thymine. Adenine will be complemented by uracil. Now, the question is, why both the strands of DNA are not copied during transcription, that is done during replication? So now, this has 2 reasons. The first reason is, if both the strands of DNA act as a template, then they would code for RNA molecule with different nucleotide sequences. So note this point, very important. To understand this, let us consider a DNA which is having the sequence ATT, CC, GAAT. So any other sequence also you could take. So the complementary strand will be TAA, GG, TTA. Now, if this strand, the strand which is I have written above, if it code for RNA, then the sequence will be this UAA, GG, CUUA, and the below strand of DNA act as a template, then the molecule of mRNA produced will have this sequence. Now, if you observe both of them have different RNA sequence. Now, if the RNA sequences are different, then definitely they will code for different types of protein. From Principles of Inheritance and Variation chapter, you have studied that one gene, one protein. So one gene will always code for one character. Character means expression of character means production of protein. Protein produced will decide the character. If one gene produces two proteins, then what it will indicate? It will control two types of characters. So if both the DNA strands act as a template, then it will code for two different types of RNA, which in turn code for two different proteins. So it would complicate the genetic information transforming machinery. So that is a very important reason. Second reason, here I have written one DNA segment, that is we written earlier also. If both the strands act as a template, there is production of two RNA molecules. If you observe clearly, these two RNA molecules produced from both the templates, if they are acting as, then they will be complementary to each other. That is, uracil is complementary to adenine and guanine is complementary to cytosine. Of course, if the complementary strands stay together, definitely they will form a double strand. If there is a formation of double-stranded RNA, then the double-stranded RNA will not code for protein. That means it will not undergo the process of translation. So, if both the strands act as a template, then complementary RNA molecules are produced which will form a double strand. This will prevent the RNA from being translated. Then the process of transcription become futile. In your text they have mentioned the term futile. So futile means the process did not able to produce a useful product. Even though if it undergo transcription, it would not produce any useful product. So these are the two important reasons why both the strands of DNA will not act as a template. Now, let us understand the structure of transcription unit. Earlier we have studied a unit of DNA will undergo transcription. So that unit of DNA is called transcription unit. So to define very simply, the unit or segment of DNA that undergo transcription is called transcription unit. Now the next question will arise is, what are all the components of transcription unit? What must be present in DNA to call it as a transcription unit? To call any segment of DNA as a transcription unit, it should possess three regions in it. They are a promoter sequence, the structural genes, and a terminator sequence. Suppose I am writing here two strands of DNA. Suppose I need to call this segment of DNA as a transcription unit. First, it should have two strands. That is for sure. And which enzyme is responsible for polymerization of mRNA from the DNA? That is DNA dependent RNA polymerase. From earlier topic, it is clear that DNA dependent RNA polymerase also polymerize the nucleotides only in 5' to 3' direction. Now, the template should have the polarity opposite to it, that is 3' to 5'. Hence, in a segment of DNA, the strand which is having the polarity 3' to 5' is called a template strand. Remember, that template strand information will be copied to mRNA. On the other hand, the strand which is having the opposite polarity, that is 5' to 3', is called a coding strand. Now the question will arise, even though it is not coding for any mRNA, then why it is called a coding strand? Carefully listen to the explanation now. If the sequence of DNA is this, suppose the template strand is having so much sequence, that is A, T, T, G, G, A, A, C, C, T. And the opposite strand of course have T, A, A, C, C, T, T, G, G, A. Now, the template will code for mRNA, right? So, the sequence of mRNA will be complementary. That is, instead of thymine, there will be presence of uracil. Wherever adenine is present, it will be complemented by uracil and remaining will be the same. So, this is the sequence. That is in 5' to 3' direction, which is coded from template. Now, observe carefully. This sequence is similar to that of the coding strand. That is, including polarity, it is same to that of coding strand except uracil in the place of thymine. So that is the reason why which information will be coded to the mRNA? The information which is the sequence which is present in coding strand. That is why it is called coding strand. And that coding strand information has to be copied to the mRNA, means which has to act as a template, the template strand. Okay. So now all the information that is reference point, so that is where is promoter sequence, where is structural genes, where are terminator sequences, all those will be defined with respect to the polarity of coding strand only. Now the coding strand 5' end is considered as upstream. So in your text they have mentioned upstream. At upstream, that is 5' end of coding strand, there is presence of a promoter sequence. So this is promoter sequence. Now downstream, that is towards 3' end of the coding strand, is the terminator sequence. Now we can switch the promoter sequence, but everything has to be switched. So remember that point. Now in template strand there is presence of structural gene. Now is that point clear? This segment of DNA is called a transcription unit because it contains promoter sequence, it contains structural genes, and it also contains a terminator sequence. Okay, so now the polarity of the coding strand will be from 5' to 3' direction. Towards 5' end there is presence of promoter, that is called upstream. Towards 3' end there is presence of terminator sequence, which is called downstream. Okay, so this transcription unit diagram is very important for the theory exams.